Hello my friends and welcome back to my channel. In today's video we'll dive deeper into the fascinating world of genome-wide identification. Especially we'll be discussing the process of retrieving genes for genome-wide identification. If you are interested in this topic, please join me so I will show you step-by-step tutorial. So let's get started. In my previous video, I explained the significance of protein conserved domain for exploring genome-wide gene family identification. So please go back to my previous video. The link you can see it in the description to get more information about the protein conserved domain and how it's linked to genome-wide gene family identification. So now I will show you some real example about genome-wide identification and how we can use it for publication. I will show you now a gene family of invertase. This gene family I have published before in PMC Plant Biology. It's very reputable journal and it's Q1 journal, and also you can see here about more than 10 papers was published related to this gene family in different plant species like peanut, like Pisium sativum, tomato, potato, Arabidopsis you can see in populus, and also glycine max, soybean, in maize. So I will show you in details more what is the information and what is the figure can be used for this such kind of publication. You can see here, firstly we need to prepare basic information about the gene family, about the gene name, amino acid, molecular weight, isoelectric point, subcellular localization, chromosome number and location, and also in the group. Actually in different genes and different gene family has different domains and has different properties. So when you are doing such kind of research, you have to read the more references to understand your gene family, and then it's easy for you to continue. And also another figure is a chromosome distribution, also like phylogenetic tree, also the gene structure, motif, structure of the gene family. What kind of experiment you can do for such kind of experiment? Just gene expression analysis. So actually gene expression analysis is very easy by some treatment and design some primer and perform qRT-PCR for gene expression analysis. And you see also this is gene expression analysis in salt and osmotic trace. You can also, there is some measurement for sugar. You can see just a few figures, and I think it's easy to do, so can be published in such kind of good journal. I will start retrieving the gene and the protein sequences of this gene family. I will give you just as an example the gene family of invertase. You can do your experiment based on your gene family. As I said before, you should read some references to understand what is conserved domain of your gene family and what is gene ID. Maybe you can get some accession number or gene ID in your references, and you have to keep in mind that there is accession number and gene ID based on the database they use in publication. So you should use the same database to be able to get a protein sequence at the beginning. But now I will explain in general by using NCBI how we can get a protein sequence as a query to find more isoforms of the gene family in our species database. So let's start it. As I know of this gene family of invertase, it has two subfamily. One is acidic invertase and the other is alkyl Newton invertase. But as I said, every gene family has some properties, and you have to know such kind of information by reading some reference. So I will introduce the name here. You can see here with some time you can get a lot of information from NCBI. Maybe if you need to explain more about NCBI tool, please write in the comment if it's necessary for you, so I can make one video for NCBI how we can use it. So I will as I said, for genome-wide identification I need to search for protein sequence. So I will search here about proteins. So now I get some hits based on this one. Maybe you can see here there is some reference sequences as transcript and protein. I will open protein sequence. You can see, so this protein it's from glycine max is a soybean, and you can see here is the locus of this gene, some information, but I would like to know to get the protein sequence. This is a protein sequence, but I need, as I told you, in every database, if I want to blast I need a specific format. So I need to use FASTA format. You can click in the FASTA. So this is the FASTA format of this protein. So I can copy and I will try to use retrieve this gene family from the tear as example. As I said, this is just example for you to show you how we can use database. So I will run blast. As I got the protein sequence from NCBI, as I am showing here, this is a protein sequence, and for retrieving members of gene family, it's better to use the protein sequence. And I select here, there is here many in plus there is several types like blast N to use nucleotide query to get nucleotide hits, and you can also use blast P amino acid query to get amino acid hit. So I will use this one, so we can blast here, and no need to do anything, just run a blast. This is one example, but based on your species you can use almost the same way. I will show you later because I'm working in citrus. I will show you how we run blast in citrus. Also I can use Phytozome because it has so many species. Also we can use Phytozome for retrieving the gene and the protein sequences of the gene family. So for example, if I want to retrieve the gene family from tomato, so firstly I can select the organism like tomato. So I use this library, Solanium lycopersicum tomato, and I search by blast. You can search in Phytozome by keyword, by gene name, or by blast. As I have the protein sequence, so I will run blast, and here there is you can enter one or more DNA or protein sequences. As I have protein sequence, so I use protein sequence. So target type is a proteome, and the program blast P. So also I can make go, so I can go back to the TER database. You can see here when I use this query, I got a lot of proteins, and you can see here 1 2 3 4 5. It has the same excision number but different transcript as shown here. This is point 1, point 2, point 5, point 4, point 3 is a different transcript, but they are the same gene because they have the same excision number. This is different excision number, so it means that it's a different gene. This 120 142 should be 3 and they are belong to actually after you are getting the sequence you should to filter the sequence based on the excision number finally to get not redundant. You have to filter that, and you should finally to get not redundant members of the gene family. How we can get the sequence? You can go down here. There is some excision number as showing here, and also you can check the affiliation. All of them is belong to soluble invertase as I type in NCBI. So showing that this by this way we can retrieve the same members of the gene family using the protein sequence. So as I use here classing max protein sequence, but I plus in Arabidopsis, and TER database is special for Arabidopsis. So you can see the homologous, the similarity is not it's not 100%, it's almost 64%. This is 63%. So however they are belong to the same gene family. How we can retrieve and get a sequence? You can open new link. So now you open the gene 83. This is the excision number. We can see here the annotation Arabidopsis thaliana cell wall invertase. So this is encoding a protein with invertase activity soluble invertase. This is more description involved in pollen development and pollen tube growth. So how we can get the sequence down? You can see here transcript. It has almost 5 transcript. You can see here more details about transcript and the gene map. We can see you can see the difference. The blue one, it's 5 UTR and 3 UTR. This is the brown is the exon. The line here, it's the intron. The different transcript, they are different. They have different exon and intron, and this is what we say different gene structure. How we can get a sequence? You can see this gene is localized in the chromosome 3, and the location this is the location in the chromosome 3. This information also you can get it for drawing the map of in the chromosome. We can see chromosome distribution. So this is the sequences that we can get for this protein. If you need the full-length CDS, full-length genomic sequence, full-length cDNA and the protein, so I will get the protein sequence of this protein. I can copy the sequence and I can open here in the desktop. You can open new file. You can see protein Arabidopsis. So we say cell wall invertase 1. The name of this protein we can discuss later about it. So you can, when we open one protein with different excision, with different transcript, we can see that the color is changed. So let's to open this protein that have, as I said, it has different transcript as well. We can see how the color could be changed. So we open now here. So we open another protein also belong to cell wall invertase. The same encodes a protein with fractalin. It has the same conserved domain. So we can go down to get the sequence protein. You can see also here it has different transcript, different exon and intron structure. So this is a protein sequence. We can copy the sequence and we can go down. We can, this is what we say FASTA format is consists of a greater sign, the gene name, or some description about the gene name. Should be no space. If you want to add more, you can add this word. So sugar, for example, more description. And this is the FASTA format. This is the protein sequence. Also, actually, I can get CDS and genome sequence, so I can open a new file. I will use it in the future. This is cDNA or CDS sequence. CDS sequence is a coding sequence, and cDNA that include UTR and exon. And this is for genomic sequence. This sequence is important to later on to make the gene structure of this gene family. So this is a protein sequence. We can go back. So for length CDS or coding sequence, we can see what the difference between CDS and copy to the CDS. This is so what's the difference between CDS and cDNA? We can see the length here from 1 2051 and go back to CDS. Also from 1 to 1751. So we use CDS that just the exon sequence. So I'll get the CDS and copy here. Save. Also I need to the genome sequence. Copy. So I can close this one. Now I go back to this gene to get the CDS and the genome sequence. So you can notice here the genome sequence is much longer than the CDS sequence because it's include 5 UTR, exon and introns. So now we retrieve 2 sequences. You can see here all, as I said, all the transcript showing here different colors, so you cannot make any mistake. So open the third one. So you can go down to get the protein. Here more information you have to, maybe you can check. Also belong to cell 1 overtase. Also showing here the function and description about the gene, so you can confirm the gene sequence you are getting is belong to the same gene family. So firstly I will get the protein sequence. Then I will get the genomic sequence. So you can see here this genomic sequence is it's about 2351, but let's see the CDS sequence should be more shorter. So it's about 1901. So now more different colors, so I can open this gene. You can see here the color are more, so I cannot get any mistake. Also you can see here the description is belong to the same gene family. So now you can see here more different colors, and you can continue by this way to retrieve all the sequences. Now I will go back to Phytozome to see what is the difference, how we can deal with this different database. This is for tomato. So this is a protein sequence, and this is the value of similarity. This is the protein IDs, more information about the location and so on. So to get the sequence, you can here view gene report, or you can view in G browser for genome browsing. So I will use gene report. So how many protein can have similarity and homologous to this protein we use as a query? 1, 2, 3, 4, 5, 6, 7, 8, and 9 proteins. So you can see here same accession number, so you have to select one of this one. Also here there is two numbers as shown here. There is two hits. Why there is two? Because they are different transcript sequence. So view this, we can view the gene. You can see here the accession number. As I said, different. Every gene has accession number or gene ID. This is the location in the chromosome. This is the description or conserved domain. You can see here glycoside hydrolase family 32. I know based on my reading in the reference, the gene family has this conserved domain, so I cannot select mistakenly any protein. How we can get the sequence? Go down here. There is different sequences. If you want genome sequence, transcript sequence, CDS sequence, or peptide sequence, it's a protein sequence. And also we can use the same protein sequence to use different database, and like in citrus we use citrus pangenome database. Also we can use here, there is several tools. You can retrieve genes based on the gene ID, or maybe by blast. We select blast protein, we select the species we want to use, like citrus. We select the database, and then we paste here the protein sequence, and then run blast, or can waste some time. So this is all the genes or proteins that are homologous and have the same conserved domain compared to the query sequence. Also, as you can see here, the same way it has this gene, like it has different transcript. So we'll select one transcript and we will remove others, like this one, it has 5 transcript. So how we can get the sequence? So different, you can open here from this way. So in this database, if you open any gene ID, you can see the G browse. This is the browsing in the genome. You can see here why it has this gene ID. It has two transcript. That's why you can see here two hits. You can 8890.1 and .2. The difference because they have different transcript. It's very common in the gene. So how we can get a sequence? You can open. If you want the gene sequence, you can get transcript sequence. Also, you can get transcript and also CDS sequence. So if you need here genome sequence, you can get this genome sequence. This is a CDS, protein, cDNA. So you can get all possible sequences from this way. So different database has difference in retrieving the sequence of the gene. So I hope this video to be clear for you, and please, if you have any question or any topic for further videos, write in the comment, and please like and subscribe